Hello and welcome to this design practice 2 module 15. I would like to today go into some more details about sensing or how sensors are designed or they are carried out, and the first important significant thing that I would like to mention are about the methods of immobilization. I think I have in great details explained to you how a sensor surface is modified by putting a recognition element or a layer which would be associating itself with the analyte of interest that you are diagnosing or sensing. And therefore, a very important premise here is how do you immobilize the recognition element on the sensor surface. And particularly when we are talking about a transducer surface which is able to monitor chemical, biochemical, biological or gaseous entities, obviously the protocol becomes very complex, and some ways and means are established for this immobilization of the recognition element. One of them is the adsorption to a surface. Then we have micro encapsulation, which actually is trapping between members on the surface of the detector. So, you have membranes. So, you have two membranes, one membrane on the transducer surface and the other after the membrane has been sprinkled with the recognition element. So, it kind of entraps, and these membranes hook themselves up to each other, adhere to each other or to the surface, transduction surface. So, this is called micro encapsulation. Then there is an entrapment process through which immobilization can be carried out. In the entrapment process, you can typically use a gel, which is a semi paste like semi fluidic kind of a state, which would be able to give diffusion, or which will be able to assist the diffusion process of the analyte of interest, and the gel typically is a network. So, you have a series of pores or sieves, it is highly porous, and it is also able to do some filtration as the analyte migrates towards the transduction surface or transducer surface. You can also entrap with paste or polymers onto an electrode. Okay, so, that is how you can do entrapment. You can do the covalent attachment of the particular recognition element. For example, you can have chemical bonds formulated between the recognition element and the transducer surface, which will give you a sense of how to immobilize. You can do cross linking. Cross linking means that you have another molecule which will put in place by means of a covalent chemistry. Okay, or because it is a bifunctional kind of a bilinker kind of a molecule. So, on one side it will link to the transducer, on another side it will have enough activity to link to the recognition element. So, it kind of holds as a linker molecule the recognition element close to the transducer surface, and so therefore, there are variety of these immobilization methods which one would use for the purpose of designing the designing the process of outlaying the recognition element onto the transducer. Then there are several other aspects of such sensors which are of engineering consequence. One of them is about performance factors, how the sensor performs with respect to how minuscule a sample it can probably take, or are there any cross reactivities which may happen, which may create a false signal. So, the performance factors, if we look at, are number one is selectivity, which is the ability to distinguish between different substrates which are there in the analyte of interest. So, substrate is basically an analyte or a type of an analyte or a type of a molecule which we are detecting. So, there are multiple such molecules which are there. We also have something called the sensitivity range, which is usually the range which at which detection can still happen uninterruptedly. So, therefore, we are talking about the least amount or the least concentration of the material of the analyte in the particular solution. So, usually sub-millimolar range is associated, but it can go all the way up to femtomolar range as well when we talk about such sensitivity range. There can be a performance factor based on accuracy, how accurate the sensor can be to pick up analytes of trace concentrations. The accuracy better than plus minus 5% is always considered to be reasonable accuracy for a sensor to qualify. The nature of solution is a very critical aspect when we design sensors. Sensors need to be designed for the chemical reactivity, the conditions such as pH, for example, the amount of abrasion that will happen, So, the sensor needs to take care of that issue, so that the transducer does not dissolve away into the solution. It also takes care of issues like what could be the temperature range at which the performance or the functionality does not change of the transducer, or for example, what is going to be the ionic strength which is going to measure. Performance factors will also include a very important aspect which is about the response time. Typically for gas sensing applications or biosensors, the smaller the better, but for the biosensing particularly, this level is probably slightly high. With the modern technology, you can say about 30 seconds or more of sensing time is considered to be quite reasonable. There is also a recovery aspect, that once the sensor surface has sensed something, whether it can come back to the baseline signal, so that it can start sensing another analyte or another solution or another sample, and for that you need to find out how much time is elapsed before, and what are the protocols that are to be carried out, if any, for a surface to again recondition itself, which again measurements can start taking place. So, time elapsed before the sensor is ready to analyze the next sample, that should not be typically more than a few minutes. pH sensing in this particular matter is a pretty commonly used electrochemical process, where we dip the electrodes in water and try to make the pH come to 7 again before the next sensing round can happen. And so, it has to be allowed sufficient time for whatever hydrogen ions have crept inside the glass bulb, the chalcogenide based glass bulb, so that it can diffuse back and it can neutralize to the 7 pH. Okay. So, some time needs because of that mechanically driven diffusion process to happen completely before the next before the sensor is ready for the next run. So, then we also talk about working lifetime of a sensor. So, stability of the, for example, the selective material, is it, for example, in particularly biomaterials like enzymes or antibodies which are used by some sensors, the shelf life may be as low as 3 months or 2 months. So, before that it is better to be used, and after that one should discard, because otherwise it will start giving false signals. So, therefore, the working lifetime also forms a very critical function when we talk about such performance factors related to the sensors. Let us now also look at one of the reasons for why we must focus on making these sensors small and miniaturized. We know that the rapidity and the sensitivity, both these factors are really dependent on the level of the miniaturization that must occur. At the same time, the robustness factor may go down because of the small nature, physical nature of the sensors when we talk about miniaturization. And generally there are many reasons for doing miniaturization when we talk about sensing in the chemicals or biochemical processes, and one of them is reducing the sensor element to the scale of the target species. So, we must understand something, that when we are detecting a small cell, let us say for example, a biological cell which is about 20 microns, or a bacterial cell which is about less than 10 microns, and we are doing it with a surface which is probably a few hundred millimeters, then obviously there are issues related to how this surface would interact with such cell, and there are going to be its own inertial delays, because the surface is extensive in terms of the sizes as compared to the target of interest, as opposed to if we were to develop a surface which would be exactly similar to the dimension of the particular species, the inertial response may not be that big of an issue in the signal that the sensor provides you. Okay. So, therefore, you can provide a higher sensitivity, and that is outrightly the case when we talk about miniaturization. Obviously, when we talk about chemical biochemical processes, reagents are very expensive. Diagnostics, particularly clinical diagnostics, becomes very expensive because of the size of the reagent, and if we are talking about miniaturizing the sensing element, it automatically means that the reagent volumes also get miniaturized, and so the associated costs of doing diagnosis by doing designing sensors would also come down because of such reduced reagent volumes. There is also reduced time to result due to small volumes, because you can have effectively higher concentrations, very small volumes, small changes being sensed by a sensitive instrumentation setup, and so any small signal which is representative statistically of the sensing modality would indicate that there is analyte present or absent. Okay. So, this is a big advantage of miniaturization, that you can actually work at those scales in much reduced time. There is also amenability of portability and miniaturization of the entire system. Obviously, smaller size, the better it is to carry off. There are concepts where sensors are taken to the bedside or even to the fields for doing detection, be it agricultural sensing or be it patient based medical sensing or even sensing of farm animals or farm products. If you can make the sensor small, it is always going to give you more portability, more usefulness in terms of taking it to the actual spot and doing measurements quickly and rapidly. This can be used more as a screening data, and based on it you can see whether you can take countermeasures then and there, rather than waiting for sensing data to come from a sophisticated lab. So, this is also known as point of care diagnosis. Okay. So, it is a very big area of work, and right now there is billions of dollars of research spent in designing and developing point of care medical biomedical sensors. There is also a scope of multi agent detection. When we talk about miniaturization, smaller the size, the better it is. You can have in a smaller space or volume many sensing elements which can sense each of these species which are there in an analyte. So, if you wanted to do multi analyte scanning and sensing, the best idea is to again start miniaturizing it and then obviously it has potential for use in vitro as well as in vivo capacity. And so, these are some of the reasons why we must miniaturize sensors. When we talk about sizing them down and we talk about small overall volume within which the sensors would be occupied, a major issue comes up, and which indicates how do you handle such fabrication, or what are the kind of agents or what are the kind of techniques which are available through which you can carry out those fabrications. So, I am going to now go across certain small modalities where we can talk about little about micro machining or little about photo driven processes through which machining can be carried out, and you can see that how small sizes can be embedded or MEMS can be made with respect to all these processes. And typically once you have an idea of how MEMS can be done, then MEMS designing, development, sensorial design through MEMS route etc. becomes easier. And for a designer who is into the area of sensors design, one must know these basic modalities when we talk about sensor technologies. So, let us talk a little bit about device fabrication. MEMS and NEMS processes typically emerged from silicon and associated silicon processes, because microelectronic industries had silicon as the base material. The carbon electronics, which is grown probably much later, did not quite come into picture when microelectronic processing was developed ab initio, and therefore, MEMS has all sort of focused onto silicon based structures, but later towards the 80s changed gears and went into the polymeric structure. So, I am going to, from my perspective, just give you some idea on fabrication on the silicon and some idea of fabrication of the polymer structures. So, MEMS and NEMS are typically fabricated by formation of structures that could be used to form sensors and actuators at that scale, micro and nano scales. Also when we talk about MEMS and NEMS, we mean that it is about the processing of electrical or non-electrical signals, non-electrical for example mass based or in thermal means, which is not driven to any electron flow as such. So, then when we again talk about MEMS NEMS silicon fabrication, we generally use conventional and new semiconductor manufacturing techniques. Some of them may be etching, deposition, photolithography, oxidation, epitaxy, and I am going to do some of these techniques with you guys, so you understand about how these techniques are being done. And then there are some which are actually MEMS specific techniques. For example, this deep reactive ion etching can be a MEMS specific technique which is used for developing high aspect ratio structures on silicon surface, or thick plating, which is related to again electroplating of metals particularly, which can be several microns in thickness, and it can be used for purpose of fabricating MEMS structures. So, when we talk about developing a miniaturized superstructure for MEMS, we mean by developing this structures which can be either bulk micro machined or surface micro machined, and there is some a fundamental understanding to what is a bulk micromachining activity or what is a surface micromachining activity. For example, if we look at the schematic here to the left, we see that this is a silicon wafer, okay, which is a thickness of the silicon wafer. This T right here is the thickness of the wafer, and we are seeing material being taken off subtractively from different regions of this particular thickness of the silicon wafer. So, any such process which involves subtractive machining where material is taken away at the micron scale or the microscopic length scale is better known as bulk micromachining. Similarly, if on the other hand, if we develop features and structures on the surface rather than subtractively taking it by adding the material. For example, if we look at this particular cartoon here on the right side, we are talking about films which are being developed on the surface through multiple techniques like deposition, etc. This, for example, is a P double plus film, okay. And then there are certain techniques which are again further used for bringing out structures and features on the surface by adding. The bulk here is not getting sacrificed or the bulk is not getting machined. So, these are the surface micromachining. So, all additive processes are categorized as surface micromachining, all subtractive processes are categorized by bulk micromachining. If I looked at such structures and how they are formulated, this structure right here can be formulated through etching. There is a process called etching, okay. Etching can be again a high aspect ratio etching which is done using usually gas plasmas. This structure here is a high aspect ratio etching. This is a low aspect ratio etching, but this can be done on a wet chemical stage with wet chemistry processes. This right here, the green region, is a P double plus silicon which is done by doping of the silicon structures. P double plus meaning thereby that you have high amount of doping concentration of the group 3 material which will pump in more number of holes. And this structure right here is basically the structure which is again etched out from the silicon, okay, and this meets the green surface right about here and proceeds no further because whatever etching is being carried out stops wherever there is this P double plus layer, okay. So, this is again a process which is called etch selectivity driven process, so that you can obtain a very thin film, a small film on the surface of this particular silicon wafer. Similarly, quite interesting in this particular case, probably there was some material which was here in this gap which has been later on sacrificially removed. The material was earlier patterned on the surface and there was a deposition of a thin film which is this hatched area right here which took place, and later on this material was removed through chemical etching, so that you have this gap created which is like a small embedded chamber which can now be used to carry fluids, etc. Okay, so this again this deposited material is a sort of a polycrystalline silicon material which is higher in strength, mechanical strength aspect. So, the idea is when this layer is removed here, it should not bend or warp, okay, the structure should be stable, and this is through the again surface micromachining that you can build up such structures. So, that is what devices are typically fabricated by. And now we will start looking into some of these processes in some little details. Let us talk about this particular process here which is borrowed from a microfabrication, a very famous microfabrication textbook. It talks about again using a etch stop layer and a certain masking process which would actually lead to the removal of this mask. The mask is the literal meaning of the English word mask, meaning it kind of shields, okay. So, wherever this masking layer is present, and I will talk a great more details about what really is the masking layer and how you will be able to remove the layer or make the layer come up in certain selected regions. So, wherever the masking layer is present, whatever chemical we are going to etch or use to etch the particular material silicon here, which is the white colored material here, okay, it does not touch the silicon if there is a masking layer. So, let us say in this region the chemical will not be able to get in touch with the silicon which is inside, and wherever there is an exposure like in this region there is no masking material. So, it will start the etching process because the etchant has already contacted the particular surface through which there can be chemical removal or subtractive machining of the particular area. So, you can carry out etching in many ways. You can carry out isotropic and anisotropic etching. This is an example where anisotropic etching is carried, and these etching happens because of a redox nature of the chemistry which is involved, and ultimately it merges as an array of planes, okay, which are corresponding to the 111 direction. Those are the slowest electron releasing planes, because of which you can have this kind of a shape. But whatever it is, without getting into those details of chemical etching, I can say that because the masking layer was present and later on you pull out the masking layer, you see there is no masking layer here. So, you dissolve this layer in a suitable solvent, but till and until it was present, the chemical did not affect the regions where the masking layer was shielding. Okay. So, this is a very important essence. So, you are covering something with a layer which is otherwise sacrificial layer, and you are using a window created in that sacrificial layer to do selective etching. So, supposing today if you were to etch on a very small region, let us say 100 micron by 100 micron pit is to be created. So, you can actually pattern and open up vias in the masking layer in a manner where exposure would be there to the parent material and the etching would sit on that and start etching. Okay. So, it is a very interesting way of carrying out machining at a very small scale, all of which can be used into what we know as MEMS, okay, or what we know as microstructures, which can then go into sensing and diagnostics. So, this again is a structure what I described to you before. There is a layer of material which is otherwise mechanically robust, but there is a silicon polysilicon layer deposited on the top of this sacrificial layer which you are removing, and so out props this cantilever like orientation of this particular material. So, this is how you do freestanding structures, okay, through microfabrication technique. So, when we talk about the materials which are mostly used for microfabrication, mostly the materials which come in handy are silicon and other microelectronic materials. The chips or the microchips which are being formulated would have the necessary modalities of being able to be transparent on the top. So, you can have covers which are made using glass or quartz which do not absorb characteristic wavelengths, okay, and gives gives a leeway for the detection process to happen of what is within the chip as opposed to its own component which it might otherwise give in the signal if it is a non-transparent or a slightly translucent kind of material. Obviously, people have realized that when we talk about clinical diagnostics and biofluid samples, they are not very well to do with inorganic material like silicon, because most of it would not behave in a proper manner as they do within the human body. And so, therefore, polymers are another class of material which have been found to rhyme and rhythm with that aspect, and therefore, people have actually developed a series of polymers which are fabricable at the small scale or the micron scale. Some examples could be PDMS, polydimethylsiloxane, PMMA, polymethyl methacrylate. These are the polymers which are used for MEMS fabrication, okay, fabrication at the micron scale, using resolutions and tools with resolutions which are high, particularly tools which would provide high resolution would be able to act on it and quickly produce an array of structures within these surfaces. I am going to get into the precise details a little bit later. Teflon, for example, is another such material, and then there are also materials which are related to the entities themselves. For example, you can today develop devices with DNA printing or where sequences of molecules are heaped together or stacked together with processes like microcontact printing or dip pen lithography. So, you can actually be able to print molecules and do some sensing diagnostics of other molecules based on the chemistry of the molecules that you have printed. So, you can make assays, okay, or assay technology in a very rapid manner to screen off a bunch of different molecules which may be desirable or undesirable in a certain fluidic sample. So, I think I am going to close on this particular module now, because I have talked enough about the basics of fabrication. In the next week's module, we will start with some fundamental processes. We will also do some aspects of actuators and how to design some of these sensors and actuators. As of now, thank you very much for being with me on this.